Welcome and thank you very much, members. I can see the registration is almost up to 60 just within 1 minute, and we expect more and more participants as we dive in. So thank you very much, the Joint Clinical Research Center. This has actually been made effortless within the webinars. This is our 6th series as the Joint Scientific Committee. We have the JCRC, we have MUHAS, we have Muhimbili, we have the National Drug Authority, we have UNCST, and our coordinator, the University of Coimbra, who have made all this possible through the EDCTP Global Health Initiative. Allow me welcome our guests today. I guess this side is actually afternoon. It's a warm afternoon in some parts. Within the city, it's actually cold. Our guest is an associate professor, Karen Dube, with the University of California, San Diego School of Medicine, Division of Infectious Disease and Global Health. She is a senior social behavior scientist and experienced research program manager who integrates biomedical research, social sciences, ethics, and patient management around HIV-related research. She has over 20 years of experience in infectious disease-related work, including the social behavior sciences and ethics of HIV cure-related research, HIV prevention, HIV treatment, Ebola, malaria, herpes simplex virus, and most recently, hepatitis B. She brings on board expertise in social behavior work and ethics to discuss ethics, access, equity, the bigger conversation. Thank you very much, Karen, for making it. Over to you.
Thank you so much, Beth. And I would like to thank the organizers for this invitation to present, in particular, Dr. Cici Kitio. I have learned so much from the Global Gene Therapy Initiative in the last 4 years. I'm incredibly grateful for these conversations. And so today we'll discuss ethics, access, and equity, the bigger conversation in cell and gene therapy. So what is ethics? Ethics are guideposts. And in our GDTI calls, we talk a lot about regulations of cell and gene therapy. But ethics are actually quite different from regulations. Ethics are principles of what ought to be done. They're rooted in fairness, justice, respect for persons, beneficence, and non-maleficence. They guide decisions when rules are absent or unclear. For example, should trials prioritize communities most affected by HIV and sickle cell? Regulations, on the other hand, are enforceable rules, and they tend to be more country-specific or region-specific. So here we also distinguish between medical research and public health ethics. In medical ethics, we care for the patients, and we have 4 core principles: autonomy, non-maleficence, beneficence, and justice. In research ethics, we care for trial participants. And in public health ethics, we care for populations. We can see where these principles can sometimes overlap, but they can also sometimes be in conflict. For example, in HIV cure-related research, where we do analytical treatment interruptions, medical and research ethics can be in conflict. So we have to recognize these different tensions and see how we can resolve them. And so if I'll make one point today in my presentation, is that issues around fairness and equity start very early on in the translational research pathway. So as soon as we start thinking about cell and gene therapy products at the bench in the laboratory, because everything after that will affect what we do downstream. So for people who say, oh, let's worry about efficacy now, and then we'll worry about equity and access later, I think this is a wrong paradigm. The correct paradigm is to incorporate ethics, access, equity considerations very early on in the translational research pathway. And this is the approach that the Global Gene Therapy Initiative has decided to follow. And it is the way forward to make sure that our products or interventions achieve maximal global health impacts. So I'll start from the very beginning of the translational research pathway, and then I'll work my way up. So what are some of the challenges with preclinical studies? So these are studies at the bench with animal models. We know that there are physiological differences among species and representative models. There could also be some methodological flaws around the lack of randomization or concealed allocation and blinding. We also need to make sure we have adequate statistical analysis and reporting. We need to report missing data or negative data. There's also a bit of a publication bias. And we also know that animal species can be biased and can vary across disciplines. And I can make any of these journal articles available if that would be of interest as well. So how can we then improve the predictability of animal studies? There are many ways to improve internal validity, external validity, and construct validity. For example, the choice of our sample size, how we randomize allocations, how we blind experiments, how we match models to human manifestation of a disease, for example, sickle cell or HIV, and so on. With external validity, this is when we want to make sure we can generalize the data and replicate in different models, for example, of the same disease, or have independent replications, or to replicate in different species. So lots of issues to consider here. And once we start moving these products into first-in-human trials, we know this involves a translational research process. So when we first test novel intervention in humans, that's when we reach a really important milestone with our cell and gene therapy product. And this is where we have to really focus on evaluating safety. What are the risks of this intervention? And when we start to determine the dose for subsequent trial. So what is the maximum tolerated dose for the experimental intervention? We do not want to cause any undue harm. In these early phase experiments, we also know that there are very limited direct benefits for the participants that are expected. Most of the benefits are societal. And so we have to find ways to have a trade-off between potential safety and efficacy. And then there's another category, which is uncertainty, which is different from risk. And that has not really been well characterized in research ethics, but we need much more conversations around uncertainty, particularly, for example, in HIV cure-related research, when we have a potential of a viral rebound, there's a lot of uncertainty about the future trial outcome. So we'll come back to some of these concepts as we move to the presentation. So lots of ethical issues in translational research for cell and gene therapy. And we know that there are many products that are expected to be approved. This is a very rich time in the field of cell and gene therapy. So what you're doing right now is on the track, because there's so many different innovations in this space. And the future of these therapies is really in Africa as well. In the next 10, 20 years, we're going to start to see the deployment of several clinical trials and novel cell and gene therapy, thanks to the leadership of Dr. Sisi Kitio. So what are some of the ethical challenges of cell and gene therapy? There are many. We've already talked about preclinical research. We also need to assess the risk to benefit profile. We need to look at how we include participants, how we provide informed consent to them. We also need to look at potential risks of these interventions, for example, immunogenicity and so on. And so we'll go through some of these different ethical considerations for cell and gene therapy products. But we know this has a very long history. There have been many news regarding cell and gene therapy. The most famous is probably the death of Jesse Eltinger in 1999, a patient at the University of Pennsylvania. And so that has sort of slowed down the field of cell and gene therapy. But now, 26 years later, we really see a revival, and this has really become very popular. So what are some of the ethical considerations for cell and gene therapy? The most important one, or one of the most important one, is favorable benefit to risk. And these benefit to risk assessments can be sometimes challenging to do. And here we want to make sure that we're doing societal good, that researchers minimize the risks and maximize the potential benefits. We also need to protect any participants from excessive risks. but these are very difficult to evaluate because of the asymmetries between the benefit and risk. And there are many other things to consider in evaluating risks. For example, the innovativeness of the intervention, how much has it been tested in the past, the mode of action, the nature of the target, the relevance of animal models and preclinical evidence. And now there's a push with the US National Institute of Health to reduce the reliance on animal models. And then uncertainty distinct from risk, which is a major hallmark of first-in-human studies, which we've already sort of discussed. And so gene therapy researchers have sort of looked at how strong does the evidence need to be in order to move a product from a cell to an animal model to eventually a human. And we know that the strength of evidence, of preclinical evidence needed sort of varies, but basically we need to make sure we have larger animal models are usually more predictive of what would happen in human. So this is sometimes a little subjective and depends on the experience of the researcher. But for every product, the investigators will compile what is called an investigational brochure that will include all the information known about this product to move them forward. So one of my inspiration in ethics has always been Ezekiel Emanuel for his landmark paper in the year 2000 on what makes clinical research ethical, the 7 requirements for ethical research. These are social and scientific value, scientific validity, fair selection of participants, favorable risk to benefit ratio, independent review, informed consent, and respect for potential and enrolled participants. And then in 2004, he wrote a different paper, adding an 8th principle, that of collaborative partnership. And this is where the issue of equity and access and capacity strengthening really comes in. We need to develop partnerships with researchers, makers of health policies, and communities in the different countries. And this is the approach that GDTI has taken. So we've already talked a little bit about risk-benefit assessment and justification. We can just appreciate how complex this is because there are different types of benefits. There are the direct personal benefits. These are the clinical or medical benefits. But in early phase trials, these are expected to be minimal. And the use of psychosocial benefits are sometimes debatable because they're really difficult to quantitate. But we know that participants benefit psychologically from being in these clinical trials and contributing to broader science. We also know people have direct or collateral benefits of being in these trials, for example, medical clinical monitoring. And there are many aspirational benefits to society and patients. So as we continue to move products into people and evaluate, I think we'll need regulatory strengthening as well to be able to evaluate these different modalities. So the IRBs will have to look at potential risks, potential benefits, and need to make assessment for all these cell and gene therapy products and also gene editing products that will be moving forward. And so this is an example of how researchers have looked at safety and efficacy and risk benefit for an AAV product, and where here the antibody gene delivery was found to be, the disadvantage were much larger than the potential advantages, so the IRB decided to not move forward with testing this intervention. But for every clinical trial, there should be a similar assessment that is done. Other literature has really looked at risk mitigation, and I really like that because this approach is really very well thought through. This is for human genome editing in the clinic. So here, the scientists have looked at the potential challenges, for example, potential for off-target activity that would result in insertion of deletion of mutations, or other potential risks like overimmune reaction. We see this with chimeric antigen receptor T cell with potential for immune overreaction with the cytokine release. Then the regulatory feasibility to overcome this hurdle, and then really clear approaches for how to address these challenges. So as we write clinical trial protocols, let's make sure we have our risk mitigation approaches that are very clear, because some of these interventions may be risky, but with risk also comes the potential for efficacy. So I mentioned this is a long history. The most recent scandal probably happened in 2017, 2018 with the gene-edited babies. I think this is a pretty famous example where He Young-Kui, a professor, used CRISPR-Cas9 technology to edit genes for HIV co-receptor in human embryos for implementation. So why was this controversial? This was controversial because a line was crossed in terms of somatic versus germline editing. The experiments edited the germline, and that's started a whole field around ethics and really needing to be a little bit more cautious moving forward. And in this case, the risk clearly outweighed the benefit because of the alteration of the germline. But there were other potential risks. For example, the incomplete ability of the CCR5 gene deletion to prevent HIV acquisition. And some of these experiments were rather secretive. And we know that the deletion of the CCR5 gene could increase risk for other viruses, for example, West Nile. And there's other much safer ways to prevent mother-to-child transmission of HIV. And so now the WHO has looked into the ethics of human genome editing and how to do this safely and ethically. My colleague, Michael Luella, who also is on the Global Gene Therapy Initiative, and I have also started looking into the ethical and practical considerations for cell and gene therapy towards an HIV cure. And we wrote a paper also with GDTI a few years ago that outlined some of these considerations. So for example, we need to have safeguards for developing cell and gene therapy approaches and safeguards for combining these different regimens. So if we combine these different approaches, that increases the risk, but that also increases the potential for efficacy. So here we have to be worried about potential harmful combinational or synergistic effect of these different interventions. And there are many guides from the regulators on how to do these types of clinical trials. So early on in the process, I would also encourage you all to consult with your regulators to find out what the regulations are in the country as you plan these different clinical trials. So also we need to mitigate potential off-target effects. And one really important aspect of these intervention is the irreversibility of the intervention. So we know there's a long-term duration of these modalities, for example, chimeric antigen receptor T cells. In the United States, people who participate in these clinical trials have to be registered and followed for 15 years. And so we have to make sure that these systems are in place. A wonderful ethicist is named Jonathan Kimmelman, has also done amazing work with these different combinations, mostly in the field of cancer, but that also inspires us in the field of sickle cell or HIV cure-related research, where he looked at when we do research, we don't really know what the target effect threshold is. We are doing the research to find out what dose, for example, will be acceptable or will be efficacious. And so we're always trying to align our target and our ethical effect threshold. And this can be very dynamic because there are many dimensions in intervention ensembles that we try to align. And these are different examples from different fields, but these are different dimensions. So the treatment dimensions are the dose, the schedule, the administration, any co-interventions, for example, conditioning, how we mitigate the risk, and the timing of delivery. Population level dimensions include like the diagnostic criteria, potential contraindications, and then the age of the people, endpoints, durations, and different outcome considerations. We also need to consider when we plan our protocols, not just the disease itself, for example, HIV or sickle cell, but also what are our clinical or surrogate endpoints. So sometimes surrogate endpoints should not be projected or perceived as benefits. And then we have to be very clear what our endpoints are. But sometimes this can be very confusing. For example, in the field of HIV cure-related research, we do not have a good biomarker to predict viral rebound. And so we do analytical treatment interruptions and we look at the effect on the viral load, but we still do not have a really good biomarker. So, and we still need to figure out how is this going to benefit the patient participants in the long term? What would be the clinical benefit? And so we have to remind ourselves where we are in the translational research pathways, what are our surrogate and our clinical endpoints, so we can plan studies that are as ethical as possible. Another issue we need to consider are social value. What is the social value of this intervention? And with GDTI, we try to plan studies that will benefit population health in the future. So there's a clear argument for social value there. And our safeguards there are the accurate assessment of the anticipated social value of the intervention, the transparency of the data, and the potential for direct benefits in first-in-human studies. So we need to see at least a therapeutic dose, or instead of a subtherapeutic dose in these different trials. We need to know that we're having an effect if we're going to go through the trouble of planning a study. But more and more now, ethicists have also started looking at, we need to think about efficacy, not just safety and social value early on in planning clinical trials. So for example, the 3 questions to assess the potential for clinical promise are, what is the likelihood that the drug will prove clinically useful in the future? Assuming the drug will work in human, what is the likelihood of observing the preclinical results in human? And assuming the drug does not work in human, what is the likelihood of observing the preclinical results? And so lots of issues to consider as we plan these clinical studies. There's another word called equipoise, and that means if participants are to be randomly assigned, this involves a randomization to an intervention, there must be genuine doubt about which intervention is better. But equipoise is much less applicable in first-in-human clinical trials. It becomes much more relevant with larger trials when there's a placebo group to look at and we compare different interventions. Another topic when we consider access and equity is translational ethics. So mapping the moral terrain of ethical or clinical research. And here, the authors have taken more of a scientific orientation to this issue of access and equity, looking at how experiments are structured across the continuum of biomedical research. For example, from planning experiments at the lab and then with animal models to moving experiments into human studies and then into application for global and population health. So at each step, they outline sort of ethical considerations at every step. This is a beautiful paper looking at how, for example, we can minimize risk, have independent review. We make sure that we respect persons and communities. We really have a lot of foresight into the downstream implementation of these interventions on a global scale. But we also need to make sure that we select potential participants adequately for these trials. And that can sometimes be challenging. Who are going to be people who can participate in these trials? Do we need otherwise healthy volunteers, or can we have people with multiple comorbidities? Would we do a trial with a person with sickle cell and HIV? So all these ethical issues need to be resolved, and they must be grounded on both scientific and ethical principles. But sometimes otherwise healthy volunteers will provide the quote-unquote cleanest data and can tolerate adverse reactions better, but they also have more to lose. And so we have to weigh many different potential risks and benefits and considerations here. This is a great example of how this was done in the field of hepatitis B, where the colleagues have looked at how do we prioritize patient populations for our study? Where do we start in terms of, and this also is relevant to equity. Do we start with those who have the highest need and can gain the best from the effective therapy? Or those that are easiest to recruit? Or those who would have fewer safety issues? Those with the higher likelihood of a potential functional cure? So lots of issues to consider, and this can also vary from phase 1 to phase 2 and phase 3 clinical trials and beyond. This is another great application of this framework, but in a context of HIV cell and gene therapy done at City of Hope, when people look at different risk-benefit ratios based on the conditions of people living with HIV. For example, people with HIV on combination ART, how would this stem cell transplant, stem cell therapy be ethically justified? And here they decide probably unfavorable because the risk could not be justified. But for people on salvage therapy, maybe we could do this type of intervention. Also, the issue of recruiting women in clinical trial. That's a big issue, particularly as we move clinical trials into sub-Saharan Africa, where, for example, in HIV cure-related research, women have really been, we need to really center trials around their needs. But this is a long issue as well in ethics. There's been a bit of a gendered logics of biomedical research, where women were excluded from clinical research for a very long time because of the potential risk during pregnancy. And in 1977, the US FDA recommended excluding people of childbearing potential from research altogether. But now there's been a lot of advocacy to sort of change that as well. And we need to really be thoughtful on how we include people of different genders in these different studies. In the United States, there's a push to center trials around the needs of women so that they can also be represented in clinical trials. So this is an article that we wrote on increasing the meaningful involvement of women in HIV cure-related research, a qualitative interview study. And we identified several steps in terms of how we plan the clinical trials. For example, having more lenient eligibility, preparing clear enrollment plans for women, setting very intentional enrollment goals, implementing enrollment freezes when necessary, when we don't have gender balance. But we also need to have clear implementation strategies as well, institutional level strategies, and also, most importantly, community engagement and outreach level strategies that would include peer navigators, outreach workers, case workers, how we involve our community-based organizations, how we use language, lots of considerations. And then most recently, there's also been a bit of a push for evidence-based or science approaches to harmonize contraception recommendations in clinical trials. So for example, for female versus male, how do we look at contraception requirements in these different clinical trials? This is a very nuanced issue as well. Other advocates have really looked for more from presumptive exclusions to a fair inclusion of women and women equity, particularly in situations, for example, where women would not get pregnant, clearly, then why would we have to do a double barrier of contraception? So really acknowledging the unique reality of each participant is important here. In the United States, we're also facing a crisis of equity. I think you all know. We need to have more attention to gender affirming care in clinical trials, but also considerations for advancing LGBTQIA+ equity in research. This means having meaningful data, safety, being granular, evolving identities of people, construct relevance, community-based research, intersectionality, and equity-centered research. I've mentioned informed consent briefly. This is a huge topic. It also relates to how people sort of interact with the information provided about the clinical trials. There are many challenges with informed consent because the information is so complex. Some of the informed consent forms I have seen for cell and gene therapy trials are now 40 pages. And so we really need to think broadly about how we are going to simplify this information in a way that is relevant to potential participants, to make sure they make decisions to participate in a way that is well informed without overwhelming them. And here we have required elements of the informed consent. But we're also trying to think about this in a more innovative ways. How can we innovate the consent? So lots of challenges and potential ways to overcome. We need to really make this meaningful to people by simplifying documents, assessing comprehensions, using more simplified, more engaging materials. We could even use audios, decision aids, and so on. So this applies to sickle cell and HIV clinical trials. There's still a lot of work that needs to be done on this in a way that is evidence-based. In cure-related research in particular, it's really important to look at potential misunderstandings as they relate to informed consent. And there are three types of misunderstandings. Therapeutic misconceptions. This is when the participants conflates research with clinical care. Therapeutic misestimation, which is when the participants underestimates the risk and overestimates the potential benefits, or both, and this happens a lot in cure-related trials. And then therapeutic optimisms, when the participants hopes for the best personal outcomes, which is less ethically problematic. Another one that we're starting to see is also curative misconceptions, where people think they will be cured when there's a limited likelihood that they will achieve cure in a particular situation, particularly in early phase clinical trial. So this is a case study of our He-Young-Kui study and their informed consent form. As you can see, there are many red flags when he did the editing of the CCR5 gene. So this is language taken directly from their consent form. And you'll see, so the research team is launching an AIDS vaccine development project. Gene editing in the embryo could knock out the CCR5 gene, decreases the risk of off-target. And there was some potentially coercive language in terms of, for example, the project team is not responsible for the risk of off-target, which is beyond the risk of consequences of the existing medical science and technology. If you decide to leave the study due to other reasons, you will need to pay back all of the costs that the project has paid for you. And if the payment is not received within 10 calendar days, the assurance of the notification of violation by the project team, another fine will be charged. And then they also took photos of the babies. Anyhow, lots of red flags. We can learn a lot from this example. Something wonderful about GDTI is the opportunity to have transdisciplinary dialogues across fields. The fact that we've had people from cell and gene therapy and HIV come together in dialogue has been incredibly generative and productive. But we can also learn a lot from the field of cancer because they've done cell and gene therapy trials for much longer. So I would encourage everyone to look into the cancer literature as well. There's wonderful examples there, particularly in the field of ethics, where we draw a lot of our examples. And when we look at different challenges and then ethical considerations for how we try to resolve some of these ethical considerations. Another development beyond the 7 requirements that I mentioned earlier, there's also been a push towards ethical criteria for improved human participant protections in phase 1 healthy volunteers, but this also applies to otherwise healthy volunteers. So expanding the ethical criteria, which include translational social value, looking at clinical research that should be designed to ensure that research results are accurate and informative for clinical populations. And this is our issue of access, that we have fair opportunity and burden sharing, fair compensation for service. This also includes our community members who provide input on our protocols, experiential welfare, where we look at how people experience these different studies. So here we know that not only the clinical harms should be minimized, but also the psychological and potential for emotional harm and social harm. And then enhance voice and recourse, giving a voice back to the participants. Also, how we use language is really important. It's an issue of language justice. From here, we've advocated for moving from subject to participants to sort of show a more active role of people who participate in research. In addition to involving our communities, there are many additional safeguards in how we plan our clinical trial. So, for example, staggering dose groups, we would not do all the doses at once. We have a small dose, a medium dose, a large dose, really important. withdrawal or stopping criteria, robust risk mitigations, which we've talked about. The crossover trial designs are helpful also to make sure everyone sort of gets exposed to the intervention, should it be efficacious. And then, of course, having very robust data safety and monitoring committees as part of our trials. It's also really important to have clear futility rules in our studies. And here the authors make a really interesting point in that if we have futility rules that are too strict, for example, on our primary endpoint, we may lose some data on our other endpoints, like our secondary or tertiary data. So how we set our futility rules, like this product not working, is also really, really important. And then there's a lot of guidance available. So this is more in the realm of regulatory, less ethics, from our regulators. So I know GTTI also considers the role of regulators in cell and gene therapy. FDA has several guidance on their website, and I'm sure Ugandan UNSCT also has guidance. So making sure we know what the latest guidance are, communicating with our regulators as we plan these trials is super important. Let's see. Another topic that's really emerging in the literature now with the availability of potential new products, similar products, is the use of platform trials where we compare different products together in parallel. So not just one intervention, but we have multiple arms in the trial. And this is helpful when we have, for example, a placebo group and we want to minimize the use of a placebo. We can have different arms, just one placebo, but it can inform multiple harm in the trial. And then more adaptive trial designs. And this has really become a dynamic field as well. There's different types of adaptive designs, like biomarker adaptive randomization, adaptive dose ranging. It's a very complex literature. The one thing I know is make sure you have a really good biostatistician if you plan to have an adaptive design. But this is where we could also move the product to more effectiveness stage. So from efficacy to effectiveness and more into implementation science type thing to really look into real world effectiveness. So I've talked about the need for a translational ethics lens to achieve equity and access. So if I have made one point during this webinar, is we need to really appreciate the continuum of clinical and translational science as we plan our intervention from the phase T0, from the targets, biomarkers, genes, pathway mechanisms, T1, first in humans, T2s, larger phase trials, potential clinical efficacy and guidelines, T3, when we really do more effectiveness work, T4, public health, and T5, global public health. But here also would like to make the case that community engagement is not just at T3, it should really be at the very start of the translational pathway, how we engage people. Let's see. And then the GDTI, of course, is leading the way in this. I've learned so much from the Global Gene Therapy Initiative in the last 4 years, thanks to the leadership of Dr. Sisi Kitio and Dr. Jenna Dare. And the approach that GTTI has taken by combining these different focus areas, like clinical readiness implementation, regulation policy, new technology development, and communication and outreach and education is so important. And it can help us not only for sickle cell, but other diseases. And there are key definitions that I think are really important to know. Justice is the fair distribution of risks and burdens. It means access to therapies beyond wealthy populations. This is where research ethics will define justice in a very narrow way in terms of fair distribution of risk and benefits for participants. But my definition of justice is much broader, like social justice. But in research ethics, we have not done a great job incorporating social justice. So we need to do better. Equity is tailored resources to achieve fair outcomes and extra support for undersourced settings. Diversity is the inclusion of biological, social, and cultural differences and ensuring treatments are relevant globally. Inclusion is actively engaging underrepresented voices, and patients and communities are involved in decision-making, so in the decision and accessibility is removing financial, geographic, and structural barriers and affordable, scalable, locally deliverable therapies. There's been great literature on this. This is really emerging. This is a wonderful paper that was published, I think, 2 years ago on scalability considerations for ethical translation in the field of sickle cell in Africa. But in this article, could actually replace the word sickle cell with the word HIV, with the word hepatitis B. A lot of these principles would still apply. It's a really great paper. It was done very well. And then including bounded justice, looking at inequities in global access, and developing governance regulatory mechanisms that will enable countries to compete equitably in the global gene therapy enterprise. So lots of issues to consider. We've talked about our translational ethics lens. These target product profiles can also be very helpful. And we know in the field of HIV cure-related research, the target product profile for a globally scalable HIV cure is cell and gene therapy. And so that's wonderful, and we need to work towards that. And these articles can be very helpful in setting, what are we working toward? The Global Gene Therapy Initiative has also published a lot of wonderful papers in the last few years on translational considerations. This is a paper led by Kevin Dockson on the translational gap for gene therapy in low and middle income countries and how we can overcome that by having collaborations between academia, governments, participants, and then commercial sphere, and how we can incentivize and so on. There's lots to be done and a lot to learn. This is another review article on how to democratize cell and gene therapy, a global approach, that actually cites a lot of our Global Gene Therapy Initiative manuscripts, including it's a web. The Global Gene Therapy Equity is really a spider web that needs to bridge together alternative regulatory and licensing strategies, regulatory hurdles, product complexity, reducing costs at every stage, and patient centricity. A great advocacy piece, Nothing About Us Without Us, Advocacy and Engagement in Genetic Medicine, led by the community here, Olabimpe and Moses Supercharger and Dr. Sisi Kitio. This is a wonderful ways to engage the community. So Moses Supercharger and his community-based organization, Jabasa, use the analogy of the remission bus, leaving no one behind. And these analogies can be really powerful in engaging the community and making research participation meaningful. And the article also provided key actions needed to ensure global access to gene therapies. So people with sickle cell disease should be major stakeholder and co-creators of this enterprise. We also need to find ways to cut costs, develop centers of excellence, for example, the one at GCRC, and make sure we can expand access to research, but also to standard of care treatment as well. So really looking at value-based systems for pricing of cell and gene therapy and integrating this early on in the research process. So I've mentioned community a little bit. So it's important to remember here that community engagement is not recruitment for clinical research. Community engagement and community advisory boards serve related but different purposes. The CAB is usually more of a sounding board, and they require capacity strengthening. We need to meet with our CAB to review our protocol. And community engagement is more broad. There's also stakeholder engagement and patient engagement. We also know that community engagement is not research or ethics. So sometimes we get lumped into one bucket, but community engagement is really engagement. Research is really systematic evaluation with informed consent. And then ethics is more of a humanities. And then in recent years, there's also been an emphasis on looking at the trustworthiness of researchers. This is more relevant in the United States, where people say, oh, there's medical mistrust or distrust. But here, this paper sort of flips the argument on its head, looking at the trustworthiness of researchers and balancing power dynamic and ensuring shared decision-making among the different partners. This is a wonderful model for assessing meaningful community engagement, a conceptual model to advance health equity, to transform health systems for health. And there are many core principles of this equity, including trust, bidirectionality, inclusivity, cultural centeredness, being equitably financed, multi-knowledge, acknowledging different types of expertise, including the lived expertise, shared governance. It needs to be ongoing. This is not one time, co-created and co-equal. It's also important to continue assessing public attitudes of these different cell and gene therapies. How does the public perceive cell and gene therapy, for example, compared to standard treatment or innovations in therapy? So generating this and doing this often is really important to see what is the pulse on our different modalities. And then how do we increase diversity in research? This is more relevant in the US settings. And how do we plan trials with social justice in mind? In the field of sickle cell, the NHLBI has done a great job sort of implementing a readiness and resilience framework for people who participate in these very intensive clinical trials. And so we try to do the same thing in the field of HIV cure-related research, where we ask different people who participated in trials with different outcomes to come and share their stories. And so many people were willing. For some of them, they went back on treatment. Others were post-intervention controllers. Others were cured, like Adam Castillo, the London patient, or Paul Edmonds, the City of Hope patient. Others, like Lauren Willenberg, are exceptional elite controllers. So across these different beautiful stories, there are emerging themes around unmet needs for people as they go through their journey. And it's really important to adopt a journey approach to cell and gene therapy. And so that's why we're advocating the community for a readiness and resilience approach and a journey approach to cure research, sort of acknowledging what people need before, during, and after their clinical trial. But even for people right now in the United States who are cured, they may face social challenges. For example, even Timothy Ray Brown, the Berlin patient, lost his housing benefits after he became cured of HIV. And so it's really important to advocate to maintain social benefits for people who achieve cure, because even people who are cured have a lot of needs post-cure. For post-intervention controllers, for us, there are many challenges as well. We know that there are unique stressors to being a post-intervention controller. People who've been able to maintain control of therapy. They may be more worried about, for example, partner protections, may have more anxiety. So we know people have psychosocial needs. So that's why we've tried to recreate a readiness and resilience framework in the space of HIV cure. And this is also underpinned by several theoretical framework. For example, the trans-theoretical model or other frameworks, and we can incorporate social behavioral research to understand how people perceive the risks and the benefits of these interventions, how they experience, how they make decisions. And we know, again, here that the therapeutic misestimation is very common in these different modalities. We also know that if we simplify the informed consent form, people have a better understanding of what's about to happen. In terms of the resilience piece, it's the same thing. We've adopted more like a stress and coping approach. So sort of appreciating the stress and the coping mechanism. And there's now a growing literature about the psychosocial impacts of being part of HIV cure-related clinical trials with treatment interruptions. And so what are some of the unique stressors, and what are some of the coping mechanisms, and how can we design better interventions, evidence-based interventions? And this needs to be evidence-based. But one area that's still a bit on that we need a lot more work in is around partner dynamics. These are data from people who participated in an HIV cure-related trial in the United States. And there were a lot of issues around partner protections. If we stop treatment, virus can come back. And so virus can then be transmitted to partners. Here, 4 out of 7 participants faced challenges around partner protections during the ATI. They had difficulty as time progressed. They had discomfort with intimacy. It was really hard to find out if their partners were on PrEP. And then one partner took emergency PEP, providing anxiety and the need for additional support. So we need a lot more evidence-based work in this area. But also partners had fertility desires. The quote here shows that condomless sex coincided with the viremic window. But we also know that partners with HIV will need support as well because they themselves are worried about their own undetectability, their own ability to maintain viral suppression. They're also very invested in the ATI trial outcome, and they do not want to retransmit to the participants during the ATI and interfere with the ATI trial. So lots of issues to be considered. We need a lot more work around participant partner protections. That's why we're trying to engage different stakeholders and create partner protection toolkits, and advocate for really justice-informed HIV cure trials with treatment interruptions. But that's really the big justice, the social justice, acknowledging the impact not only on participants, but also partners, participants, affected community, and global HIV science. We need to do a better job bridging cure research with prevention, but also treatment as well. So I want to mention a really unique project here at the University of California, San Diego called The Last Gift, where people with HIV at the end of their life donate their tissues so we can identify where HIV persists across the entire body, including the brain and other deep tissues. And this program has been in place for the last 8 years now. But very early on, the community was engaged to generate ethical considerations to do this research safely and ethically. And the Community Advisory Board really emphasized the need for patient participant centeredness and helped us going beyond and above ethics to design considerations for people who participate in these end-of-life studies. And we've now recruited 58 people in this study. We were able to embed psychosocial assessments. Here are data from our quality of life instruments at the end of life. We actually found that people with HIV had preserved quality of life at the end of their life. We can only imagine what this was like 40 years ago when people were dying of AIDS. And so now we know that people have preserved quality of life. Something about having peace of mind at the end of life, interacting with research staff is sort of preserving quality of life. So this is giving us hope that we can continue. And one potential next step would be to introduce interventions in the end-of-life translational research model. And we're also interested in looking at cell and gene therapy interventions in this model, not only by taking cells ex vivo, for example, looking at effect on myeloid cells, but also eventually in the future in vivo. There are lots of people asking, are we having an impact in tissues? We don't know because we're not collecting human tissues in some of these trials, but this would help us also look at effect on tissues. So as we move towards ethics and towards clinical trials, either sickle cell, HIV, we're really advocating for a collaborative ethics model because when we integrate ethics, we can really push the science and innovate. This is a really great paper on innovating collaborations between ethicists and life scientists. This needs to happen not only at the conceptual stage and also the normative stage, more theoretical, but also we need applied ethics. We need to collect the data. We need this to be evidence-based. And this then can feed into the regulatory science and the legal aspects. But in my field, HIV cure research, there are many areas that need consideration as we move forward. For example, ATIs, treatment interruptions at ART initiation. We know people have a lot of psychosocial needs, placebo-controlled trials, extended treatment interruptions, multiple treatment interruptions, or individuals' ATIs, and now TTIs, tiny treatment interruptions. Research-limited settings, we need a lot more formative research. Cell and gene therapy trials in the Western world, but also in Africa. Combination trials, third-party risks. We talked about partner protection. And then interventional trials at the end of life. But once some of these categories are combined, that's when we really need really good community engagement from the very start. And we also recommend integrating social behavioral science and ethics to make sure we do not cause any undue harm and we can mitigate harm in due course. So lots of ethical questions remain to be considered here, and I really look forward to the discussion. Thank you all so much for the opportunity to share.
Thank you very much, Karen. It has been actually quite a wonderful discussion, and I see questions are still popping up within the Q&A tab. Just to start us off, you talked about intersectionality, and I wanted to find out what you do, especially with regard to class. I know we talked about the LGBTQ, but how do we handle the issue of class, given like a country or in Uganda where you have quite higher class and low class, depending on the geographical integration? How would you deal with that? And in addition to that, specific for Uganda, where we are still learning and also utilizing trials using the adaptive study designs and all that you've mentioned, that you think could be actually flexible enough given the reduction of costs and also ensuring that the participants get in innovation. How do we ensure that the trials platforms are not misused and the data obtained is actually of safe nature for the participants? Then I think with those two, we can dive in to have the ones that have been highlighted in the Q&A. I see the ICT support with the hand up.
Yes. This is Sisi.
Yes, Dr. Sisi.
Can you hear me?
Yes.
Thanks, Caroline, for excellent overview. And we have a very good moderator from one of the regulatory bodies in Uganda, Uganda National Council of Science and Technology. You've talked about mainly the ethics of actually conducting the clinical trial, presupposing we have a product, and everything you've talked about is excellent. And but the situation is there is a very big gap between gene therapy research and development, especially for diseases that are highly prevalent in low and middle income countries, and where the clinical trials are taking place. We talked about, of course, the papers that are sort of trying to see global engagement and involvement. And the ethical principles we talk about, justice, equity, diversity, and inclusion, all apply to not having clinical trials where most patients are. And you also compared ethical principles and regulations, and all these things are in regulations. But I know this is a very provocative question, but I think we have to now to go back and discuss what regulations do people or should guide these, in particular gene therapy, because that's what we are talking about, where it is developed and where it is evaluated. Because what is happening today, I think, is unethical, given all those principles that you have talked about. So this question you can attempt to answer, but I think as a global community, we have to see which regulations are there, or if there are none, what more should we be doing to make sure that we are involved in gene therapy research and development. You have given a very good slide on collaboration between ethicists and life sciences. And I think that is very important. We are currently talking about, community engagement and clinical trials, but I think we are not engaging ethicists enough. And when we go to the regulatory authorities to review these protocols, it is the ethicists, some of them sitting there, and very negative about what we want to do. So I think that is a good lesson that we have to engage in time. And the moderator talked about adaptive study platforms. These are new. They were highly prevalent in COVID, but I think we will be bringing other studies for you to review on adaptive study platforms, but I think that is something that we can seriously think about as we move forward. Thank you.
Thank you, Sisi. This is a great point. I should have showed a map of where the disease burden is versus where the clinical trials are. I completely agree with you. We need to do trials where the disease burden is taking place. And then if we do trial, we need to also ensure post-trial access. I agree with you that we need better regulation to guide this. We need better requests for applications. This needs to be built into the RFAs and the funding mechanisms. How RFAs are written in our design is so important. Where's the money going? Who's making the decision? Who's first author on the manuscript? What capacity strengthening is in place? And sometimes I find a conflict between some of the clinical trials that are, time sensitive, cost constraint versus the need for capacity building, capacity strengthening, which was more horizontal. So we need to create mechanisms like the Fogarty that are not so tied to a specific project, but are tied to building long-term capacity to be able to take a more active leadership in the future. But it's also how funders are making decisions around funding allocations and how they are designing the RFAs. And we know right now there's a bit of a crisis of this in how funding is occurring in terms of the current climate. A very great point. I appreciate you mentioning that. In terms of the adaptive platforms, I agree. I think selection of the different products in that platform is going to be really important. Lots of considerations will need to be applied there, and with a lot of engagement of scientists and community members and regulators in country and biostatisticians. So again, multidisciplinary work needs to be happening. We need to have the preclinical evidence. On the issue of class, Beth, I think it's really interesting, and I would love your thoughts and thoughts from folks in the who are currently participating. What can be done? I think we need a lot of community stakeholder engagement because that's a really important topic of equity, and it should be part of the intersection discussion.
Yeah, I think in that case, I think we would actually look at utilizing the community advisory boards more,
Yeah,
and to ensure that the feedback that they give is actually enshrined with what is actually implemented. Because I know these are going to be high cost interventions, and you wouldn't want to be it skewed towards those who can actually afford the therapy. Yeah.
I agree.
Then looking at therapeutic misconception and misestimation and optimization bias, what do you think the cooling off period would be? Because given the fact that even if you give a short informed consent that you indicated, one would actually want to give consent and ensure that the information is actually understood. And with the communities we live in, you find that if you're married, sometimes you may want to go and maybe inquire from your husband or inquire from your mom, your brother, or your sister. So what do you think ideally the calling off period minimally should be in terms of informed consent to ensure there is no therapeutic misconception or misestimation?
That's a great question. So there's always the consent at entry, which is really important. And then there could be like a test of understanding. We need to recognize also consent can be a process. There could be other people involved. I'm moving towards, for me, my dream would be to generate more evidence around informed consent using theories like fuzzy trace theories, how people make decisions, to look at across the entire journey, what are the key points where we need to intervene? Because I really think there could be micro doses of information to talk about the risks and potential benefits at specific time points. For example, in my field of HIV cure research, what we say at entry may be everything has to be there, but then before the ATI, there could be more information that needs to be emphasized. The viral rebound, we know, is also a very traumatic part of the journey. So what needs to happen there, not just in terms of informed consent, but also psychosocial support. And then when we restart treatment, what needs to happen there? What information needs to be given for ART readherence? And then at the end of the study, usually also people post-trial also want to be contacted about information about the trial. how their participation helped advance the science. So I really think it should be done across the entire journey of participation in a way that is very evidence-based. But I feel like informed consent is a huge topic. It would require reforming informed consent because I think there's some of our forms have become so long that it's really difficult to assess whether people have fully understood what they're getting themselves into. That's my personal opinion. Great point. And you made a point earlier about community feedback. I agree with you, and that also applies to consent, is if we go to the community and we ask them for their thoughts, we need to make sure that we implement whatever considerations or recommendations the community is making. So going back to the community and saying, these are based on your feedback, these are the things we could implement in the protocol. And this has to be a dialogue. Not everything will be possible because of cost or feasibility, but I think it's important. And this is not just a one-time thing. It needs to happen throughout the design of the clinical trial protocol, throughout implementation. And so incorporating the community early on and involving them in how consent is built is so important.
Very true. So we have a few questions within the Q&A. One is looking at the potential off targets that you talked about. Are there any models that we can use to try and simulate any potential off target effects before planning or before cell level experimentation? And then another goes to ask what considerations may be taken to factor in reactions of gene therapy in smaller animals that may not exactly be a risk to humans. And are the risks and benefits weighed, or they are all considered equal? I think that's the question within the chat. If you could answer those three and see how we could move on to the next. Thank you.
Thank you, Timothy. These are great questions. They're very difficult. I do not know the answer to the first question. Are there models to try and simulate the potential off-target effect at planning or before cell level experimentation? I do not know the answer to that question. I think it depends on the modality being investigated. And I think there are also assays to look for potential of off-target, and there's so many assays, for example, with CRISPR-Cas to look at potential off-target. Like the seq assays, I do not know. I would have to dig into the literature. I think it's more of a biomedical question, but it's a really good one. And not all off-target effects are bad also. So, but some are really bad, like the double-stranded breaks. And so I think this may depend on the type of modalities and also the stage of the research. What considerations may be taken to factor in reactions to gene therapy in smaller animals that may not exactly be at risk in humans? Great question. Not all preclinical models are predictive of what will happen in humans. For example, we've cured many mice, but we haven't been able to cure people, and so on. So I think just really close monitoring of these different risks at different stage is really important, and taking a scientific orientation and a risk mitigation orientation. And then are there risks and benefits weighed, or are they all considered equal? Yeah, that's a really good question that the field of ethics have been trying to answer for decades. Some risks may be more critical, like the potential for serious adverse events, I think needs to be weighed more heavily. Some of the psychosocial risks and benefits are less difficult to quantitate, but I think also have a very important role to play in these conversations. There could be financial, legal risks as well, lots of different types of risks, and these need to be part of the conversation. Thank you. These are great questions.
I think we could go. I think this is Sekanzi I think, I don't know, he wants you to comment on the milestone reached by the scientists who used CRISPR technology to completely eliminate HIV from AIDS patients without viral particles replicating again. They want a highlight on what happened.
I do not know the answer to the question. I don't know if Dr. Sisi knows. The milestone reached by scientists who use CRISPR-Cas technology to completely eliminate HIV particles from AIDS patients without the viral particles replicating again or evolving again. I do not know the answer.
Well, what is happening, there are different strategies that scientists are looking at in terms of HIV cure. And some of the gene therapy is to use immune cells that are modified, not necessarily using CRISPR, but those modifications have to be carried in a vehicle to the cells of interest. And the scientists have used lentiviral viruses, which can replicate and which can enter the cell, replicate so that they deliver. Others have used adeno-associated viruses, and those are the main ones that have been used. But there are also other vehicles which include nanoparticles, very small particles, or lipid nanoparticles that can vary, for example, the CRISPR-Cas into a specified region of the cell so that we can excise the provirus which is embedded into our DNA. And so those ones are not viruses, but for now, of course, we do not have a cure yet, but a lot of work is ongoing and people are trying out different aspects, including working on the CCR5 receptor to create a delta 32, like you already talked about, Karin, so that those cells are resistant to HIV infection. And that may be a good strategy or maybe not a very good strategy for us in these areas where we have those viruses where people may become more prone if they have this delta deletion. So there are many things that are being done or strategies, and some of these, you have to carry your gene therapy product to the cell of interest. You can use a viral vector, but others are just looking at nanoparticles, different nanoparticles, and different, to carry either CRISPR-Cas or other nucleases. So I think the race is still on, and we don't know what will be as effective. So we cannot say maybe today, don't stop this research and only continue with that. But the CRISPR-Cas has become like the frontier in the medicine of gene editing, because you don't need the viruses to take it to the cells that you need to. And now the CRISPR-Cas can be used to either to also do what we call base editing, so you do not have to cut the whole DNA. It is very exciting stuff. I'm not a virologist, neither am I an immunologist, but this is what is happening today. And of course, Karin is a social scientist, but we all read around what is happening. Yeah.
Thank you, Sissy. Beth, you are on mute.
You're muted.
Oh, sorry. I think since I have you, Dr. Sissy, I was wondering if we have done any evidence-based study on informed consent for our setting, especially to identify the cool-off period and also the need for consideration of next of kin and basically maybe spouse, wherever the person is coming from.
So that is a good research question. I don't know who has asked. You can come and join us and we do that research project, because I think we need it. We are also seeing, of course, not just for gene therapy, increasingly more complex trials have very big informed consents. And how do you ensure that there is apprehension of what you're giving to the participant? How often should we repeat this across the whole follow-up of the participant? Should we cut this because people forget and repeat certain aspects, like if you have to interrupt antiretroviral drugs when you're doing an HIV cure trial? Please, our ICT people here, whoever has asked this question, please come and work together, because I think this is very important research, even as we head to more, as we head to the hard science. And I'm sure Karine would like to be a collaborator on that.
Absolutely. Thank you.
Thank you, Dr. Sisi. I think I'll reach out on the evidence-based study. So 2 more that we have within the chat box. One wants a comment on whether the twins have been followed up, and do you have any formal communication or follow-up on their end? And then Okello Duke wants to thank you, Dr. Sisi, for what you can do, and wants to find out how more or how best we can help JCRC's attempt in bringing cell and gene therapy in Uganda. I think the last year is, what is your view on cell and gene therapy ethics in practical hurdles, for instance, in vitro models, conscription and cell therapies, manufacturing and engineering, etc. So those are 3 questions. Over to you, Dr. Karen. And members on board, if you could kindly fill in the evaluation that has been sent to you. Thank you.
Thank you, Beth. In terms of the twins, I do think there was a follow-up article. I will try to find it. I do think they try to do a follow-up on the twins. I can ask Michael Luella. He probably would know as well. And I will send that to Sisi. I believe there was a few articles published subsequent to these experiments. With regards to GCRC, I defer to Dr. Sisi in terms of what is happening.
Yeah, one more. I think she'll answer it late at the end. If you could kindly proceed to the next. Sorry.
What is your views on cell and gene therapy ethics in practical hurdles, for instance, in vitro models, cost competition with standard therapies, manufacturing and engineering, CAR T cell therapies? All really great question, Benjamin. I think as we move forward, we will face more ethical challenges. This is dynamic. This is real time. I'm less familiar with in vitro models because that's more biomedical, but I would encourage you to document your hurdles, your challenges, and how you're overcoming these challenges and publishing on it. I would recommend to do real-time ethics as part of your implementation of cell and gene therapy. This is a dynamic field, so I would document the issues you're facing, and I would issue call to actions to try to overcome these different hurdles. This is real-time ethics. It's happening in the real world.
Okay, a question on compensation. You talked about follow-up period for 15 years. What has been your experience for compensation for injury, and how has this been handled, especially for injuries that may be long-term to the participant?
Great question. This is usually spelled out in the informed consent form, the compensation for research-related injury. I think it can also vary by setting. What is compensated versus what is not down the line if there's a serious adverse event, for example. I think this can be trial specific. Also, it's important to engage with the regulators and find out what their local regulations and requirements and expectations are. And what will be reimbursed versus what will not be reimbursed. I think it can also vary by institutions. So I think it's important to know what the practices are in the different setting. I think it's very context specific.
Okay. A question is on how can you guide on reporting negative results of clinical trials, and does it circulate among the scientific community, display to journals, news outlets, probably with an example? I think Okello wants just an example of how one reports negative results.
That's a great question because sometimes scientists want to publish positive results. So there's a bit of a bias in the literature towards more positive results. There's actually a journal of negative results now where you can publish your negative results. Getting those results out there is so important. It's part of ethics as well. This prevents duplication of experiments that may not be efficacious. It prevents waste of resources. It prevents transparency among researchers. I think it's very important to publish your negative results as much as you can. But it's not easy.
Then the next, yeah, it's not easy. The next question is on how you convince individuals to participate in trials, describing to them the potential dangers. I think we've tried to answer this within the informed consent session. First of all, ensuring that the information is very short and very key to what the participant can understand, include assessment of understanding, include visuals, and the like. I think Dr. Karen Wright went the right path with that.
Yeah, I agree. There's definitely a phenotype of individuals who want to take risks in order to advance the science and to face potential efficacy. So it's also important to remember everyone is different. Some people may be comfortable staying on treatment, but others really want to see innovation in the science and help future generation. And I think sometimes people may be willing to tolerate just a little bit more risks in exchange for efficacy. Like we may need to expect an immune reaction for the product to know that it's doing something. So I think working with each person, understanding their risk tolerance is really, really important.
I think I see a hand up from Dr. Sisi.
I think there was a question regarding what JCRC is doing in bringing gene therapy to Uganda and Sub-Saharan Africa. So beyond this project, which is the capacity project funded by EU, where we are a participant with others that Beth mentioned, we did establish or co-found the Global Gene Therapy Initiative with Professor Jennifer Adair, who was previously at Fred Hutch, to try and identify the gaps that are presenting gene therapy research and development in non-middle-income countries and address them systematically. So this was set up in 2020, and the goal was to have the first clinical trial, whether HIV or sickle cell disease, in 2024. This has not happened, but we have made several strides. And Karen is part of DGTI. We invite more of you to join DGTI. We have a meeting every week on Thursday at 6 p.m. Uganda time to discuss, to invite other people who are working in this space to inform us on what is happening and then what we can do. We have, through the DGTI, have found some initial grants to train people, and some of them are already back. We have a postdoc who has done training at Fred Hutch in gene therapy manufacturing locally. Now we have a Fogarty grant, research training grant, which started on 1st of July, where we will recruit. We already interviewed the first batch of master's degree. We will also be interviewing within this month the first batch of PhDs, and the goal is to build the capacity. That is only one aspect, because the gaps are many. There is regulatory. We've been working with the Beth can tell you what we are doing with them and other regulatory authorities to get the policy signed by the president. It had gone to sleep. We resurrected that. We are part of many consortia that are educating the community. This is part of educating. This community is mainly research scientists, but we are also educating the community, lay, civil society on gene therapy. But now, today, after this meeting, I think we have to go and talk directly to ethicists, because they are the ones who might, if they don't understand what we are doing, they can make things difficult for us. But the regulatory authorities, we are already working with them in this capacity project, National Drug Authority, Uganda National Council of Science and Technology, and we are also working in collaboration with several inventors of products, because if we start from scratch, even when we get people trained, it will take a very long time. So there are already people who are willing to work with us. And one of them is an HIV product. It's a dual CAR T cell. It's being developed by Caring Cross based in Maryland. The phase 1 trial is happening. Our proposal has already been pre-reviewed by the Uganda National Council of Science and Technology, but we are in the process of setting up infrastructure to be able to do this trial, because it's an ex vivo product which needs us to develop the gene therapy in the lab. We need a GMP facility. We need special equipment. So in Makerere, they have already had some training at Yale University in CRISPR-Cas technology, and we are going to establish a training of many people who applied for the masters, but we can't take them, so that people can get introduced to these principles and have hands-on in the lab. So whoever asked this question, come and see what you can contribute. If you preferably come with a grant, you'll be most welcome.
Okay, thank you very much, Dr. Sisi. I think Timothy and Lillian, this goes to you. You've requested if there are any opportunities to participate to help JCRC bring cell and gene therapy on the map within Uganda. You have an open door by the director. There's a question by Angela saying, since UNCSC has no clear guidelines on ethical issues that arise from genetic studies, what is our benchmark in case of ethical dilemmas, such as some social implications? Allow me answer this from a Ugandan perspective. The revised guidelines 2025 on human research have guidance on genetic. There's a section on genome and genetic studies, and the UNCSC together with the partners, JCRC, NDA, are developing a context and specific guidance document on gene therapy and stem cell, on gene and stem cell therapy guidelines in Uganda. So these will be out hopefully next year. There's a question for you here, Karen. Is it possible for you to share your lovely presentation for a good read, as well as so that you can be able to also read up the references provided? You're muted, unfortunately.
Oh, yes, sorry about that. Yes, I can definitely share the presentation and the references provided. And I would encourage you all to have regular ethics check-ins or ethics huddles, where you include ethics on the agenda, and you, like, what ethical issues are we facing today, and encouraging conversations and open conversations around that, and never letting go of what makes this research ethical, and like with your experiments, but also globally, what makes this research ethical. I think it's really important to have very open conversations about this.
Yeah, very true. And I think the series such as this is one step towards the ethical hurdles within Uganda. So all thanks to JCRC for this platform. So participants, I hope we've been able to fill in the evaluation as we come to a conclusion to this wonderful webinar. Are there any parting notes, any questions before we close? Okay, I can see here litigation has become common in research. How do researchers protect themselves during consent process, especially when you want us to summarize the ICFs and the details are removed? I think that goes to the summary consent Dr. Karini talked about. Another question is there any platforms such as WhatsApp or Telegram group where further conversations are shared in regard to gene therapy research? I think it is again Sekanzi. I think reach out to JCRC. We got to be given a platform. So, Dr. Karen, can you comment on the short version of the informed consent that you had highlighted?
Yes, that's a great question. The short consent, it was an experiment. So we did experiment, we compared a long consent versus a simplified consent in an experimental setting, and I can send the paper. The short consent was actually preferred, and people retained more information about the trial that was meaningful to them, and also had more positive, effective attitude toward the study itself. So I'm happy to share that. This was done hypothetically. I think it would be nice to do this in an actual clinical trial to test simplified consent, but we need a lot more work in this area, and advocating for this is also really important.
Okay, thank you very much, Dr. Karen. I think we've come to the end at the top of the hour. I hope we have actually filled in the evaluation form. The gene therapy discussion today not only affects us as Uganda, but affects the whole global network. And we're happy to note that Uganda is one of the countries that is moving forward with the technology to see that we improve the health outcome within the country and hopefully globally. And incorporating ethics, equity, and access at the start, as you indicated, is actually very key in achieving the global impact. And I'm happy JCRC is actually leading this and has gone ahead to do some of the community studies, community engagement, and also provide training through webinars such as this. So for Uganda, and also for Uganda National Council, from where I'm from, I think we are very happy, and we're happy to note that this is going through a very systematic direction and hoping to see fruitfulness at the end of the tunnel. So thank you very much. Over to you, Dr. Sisi, for any closing remarks.
You greatly, Karina, honoring our invitation, and we thank all our collaborators in capacity for enabling this. With the hard. And hope people have filled in the evaluation form.
Thank you. I think the sound cut off briefly.
I think we're having a hard time getting you, doctor. You're off for quite some time and then on.
Can you hear us?
Now I can, yeah, we can now hear you.
Sorry, I was just thanking you for the presentation, Karin, and Beth for moderating, and I think there is still a lot of work for all of us.
Absolutely. Thank you so much for the opportunity, and feel free to reach out if you have any questions. I will put my email in the chat as well, and I will look for the article on the twins, and I will send the slides as a PDF. Thank you.
Thank you very much, Sisi, and thank you very much, Karen, and the participants for making it a wonderful webinar. Thank you.
Also, the recording of the webinar is posted on our website, so people can go back and listen. There is also a YouTube of this, so yeah.